Hello and welcome back to our third and final installment of the virus unit, and today we're going to kind of do an extension on viruses. And we talked before about how viruses were basically just pieces of nucleic acid surrounded by a protein. Well, we're going to look at two viroids and prions that are just the nucleic acid and just the protein. So we'll start with viroids, which are small 250 to 375 base pair molecules of single-stranded RNA that lack a protein coat. So they're basically just a naked piece of nucleic acid, and they become incorporated into the cell. They take over the cell's machinery to replicate and make many more copies of themselves. And they're in terms of animals, we don't really have to worry about them. They infect mostly plants, but in terms of crops, potatoes, tomatoes, cucumbers, coconuts are some examples of plants that viroids will affect and have a big impact on the crops there. Here's just a quick picture, and as you can see, here's a small circular piece, here's a little kind of a strand, and they are extremely small and hard to pick up and very different from viruses themselves. And as you can see from this diagram, the genome size, they are down here on the order of hundreds of base pairs rather than thousands or more when we talk about other viruses, bacteria, and higher eukaryotes. And because of that small size, they also have a relatively lower mutation rate. Now on to prions, which are hit a little bit more closer to home, as we've all heard about mad cow disease, and maybe in Vermont, chronic wasting disease in the deer population. But prions are basically just misfolded proteins, and as we can see from the diagram, here's the normal protein, the prion, and it basically plays a game of tag. And when that prion comes into contact with the normal protein, it changes the shape, and so we end up with areas, usually within the brain, where all the proteins are changed to a different shape. And here's an example for humans, Creutzfeldt-Jakob disease, and in animals, bovine spongiform encephalopathy, BSE, which you probably know as mad cow disease, chronic wasting disease, which is in New York State, and they're monitoring it for the Vermont deer population, as well as scabies. So these are some diseases you may have heard of that were caused by prions. And here's an image of what that protein might look like with a normal folding. So you can see the alpha helices units here, and in the prion, now we have a misfolded protein. So you can see the helice regions where the green and the orange are now a different shape, and the prion doesn't function as it's supposed to. And as I said before, they kind of play a game of tag. So here's our misfolded proteins in blue, and then we've got our normal proteins in purple. And as the prions come in contact with those normal proteins surrounding neurons in the brain, they cause them to misfold, and we end up with these areas with many misfolded proteins. And as we can see from this image with the Creutzfeldt-Jakob disease brain on the left, you can see that it looks very different from the normal brain, and there are gaps in areas where nerves have been destroyed by these prions and basically opened up. And so what we're going to look at now is a quick video that will show how prion diseases transfer and how they affect those proteins.
Prions are hypothesized as the causative agents of several neurodegenerative diseases. Prion diseases are unusual because they appear to be caused by an infectious protein. The proposed mechanism of the disease is that the infectious protein catalyzes a conformational change in a normal protein so that it becomes the same as the infectious protein. According to the proposed mechanism, the normal protein is found in neurons in the PRPC conformation. In this conformation, the protein is harmless. The infectious protein conformation, PRPSC, is introduced to the neuron after the individual comes into contact with it in their food, or in rare cases from contact with bodily fluid of an infected individual. There are also some inherited cases where it appears that the PRPC can convert spontaneously to the PRPSC form at a low rate. However, the PRPSC form gets into the neuron. Once it is there, it contacts a PRPC protein and catalyzes its conversion to the PRPSC form. Once it is converted, it too can act to convert other protein molecules from the PRPC form to the PRPSC form. Over time, all of the PRPC proteins will be converted to the PRPSC form. Some PRPSC proteins are excreted from the cell and thus are able to spread to and infect other cells. PRPSC proteins accumulate to high levels over time. They are deposited as dense aggregates that are thought to be responsible for disease symptoms affecting the nervous system.